Dear Christian, thanks for organizing this symposium. So in my talk, I will discuss the role of domain general networks for language reorganization. And I will provide examples from the combination of plasticity-inducing neurostimulation protocols and neuroimaging to investigate short-term reorganization within and across networks, mainly in the healthy language network. What do we know about short-term reorganization? I have recently suggested a model for flexible redistribution in the language network. And one core assumption of this model is that after large-scale disruption of the specialized network induced by neurostimulation or by a brain lesion, we may observe compensation via recruitment of domain general networks. So such networks may support the maintenance or recovery of function. How should that work? Let's assume we have a disruption of a key region for language induced by inhibitory neurostimulation or a brain lesion that results in a decrease of the contribution of the specialized network. Now, in some cases, we may observe a stronger recruitment and upregulation of domain general networks that helps to maintain or regain function. Domain general functions such as attention, cognitive control, or working memory are known to support all higher level cognitive processes. And the key idea here is that if the specialized network is decreased in its contribution, stronger recruitment of such domain general networks may to some degree compensate for the disruption without, of course, completely replacing it. Now, one question that I'm currently interested in is whether such short-term reorganization via plasticity across networks is a key mechanism of reorganization across different brain states, that is in young and aging and in diseased brains. And I think a better understanding of such flexible redistribution may help to develop more effective treatment options with neurostimulation protocols. So what do we know in the healthy young brain? I will first discuss targeting language-related brain regions to map plasticity across networks based on recent data from the reading network. So a first study led by Sabrina Turka from my lab, here we were interested in short-term plasticity during reading in the healthy brain. We know that reading engages a distributed network of mainly left hemispheric areas. We reasoned that if we target the left posterior superior temporal gyrus, so a key reading area, then we may decrease its contribution, which may result in compensatory short-term reorganization. So we had healthy participants perform reading tasks of pseudowords and words after receiving TMS. We observed that inhibitory RTMS over the left PSDG slightly delays pseudoword reading. Now the question is, what are the neural correlates of such modulatory inhibitory TMS effects? We observed strong inter-individual variability at the neural level and no clear pattern at the group level. So there was no evidence for changes in task-related activity at the stimulated area. And what you can see here is the RTMS effects on task-related activity during pseudoword reading plotted as overlap for at least 2 subjects. You can see that there was strong variability here. Some subjects showed an upregulation of areas mainly outside the reading network without any clear pattern or a strong overlap between many subjects, whereas other subjects showed a decrease in task-related activity or downregulation, but again, no clear pattern. So in the next step, we used a multi-voxel pattern analysis because such multivariate approaches should be more sensitive to capture the modulatory TMS effects. That is something that we have shown with different language tasks before. And indeed, the MVPA showed a shift of the task-related activity during pseudoword processing. So inhibitory RTMS over left PSDG, so in this area, shifts task-related activity towards IFG and IFS, so an area associated with phonological or general working memory processes, likely a domain general contribution here. And finally, we were also interested in changes in the effective connectivity and the modulatory influence induced by TMS. So we performed a dynamic causal modeling analysis in a predefined reading network, and we found that RTMS over the PSDG increased the facilitatory drive of the left ventral occipitotemporal cortex on the targeted PSDG. And we reasoned that this increase in the facilitatory drive after disruption of the other node likely reflects compensatory reorganization, which would be in line with previous studies from our group and others showing an increase in the facilitatory influence of another area on the targeted area after disruption that was behaviorally relevant. So in this case, the behavioral relevance remains to be shown, but we think that these results together provide some first evidence that both language-specific and domain-general short-term plasticity, so domain-general working memory areas and changes in the connectivity inside the specialized reading network can be found in response to inhibitory neurostimulation of the reading network and may help to maintain task function, may be compensatory here. Now, in the second study that is still ongoing, Sabrina is interested in short-term plasticity during reading in people with dyslexia. Dyslexia is characterized by reading impairments and often associated with an underactivation of the specialized reading areas. So we reasoned that if we apply facilitatory TMS over the PSDG, a reading area, then this may reshift the language-related activity and should be associated with better behavioral performance in this people with dyslexia. At the behavioral level, we found that facilitatory RTMS of left PSDG improves the overall reading accuracy in people with dyslexia in the task that was performed in the scanner after TMS. So they became better in overall reading of words and pseudo-words. Now, what are the neural correlates of such improvements? We found that at the univariate level with univariate analysis, RTMS decreases task-related activity in right hemispheric domain general areas during word reading. So no effects in the specialized reading network, but a decrease of the task-related activity in the right IPS and SPL domain general area. And such decreases in task-related activity likely reflect decreased task demands. This is something that we have observed during other language tasks with anodal stimulation during fMRI. If the task load is decreased due to facilitatory stimulation, we see less activity in these multiple demand areas. And here, MVPA results could show specific changes induced by neurostimulation that were task-specific, so specific for word or pseudo-word reading. For word reading, we observed shifts in task-related activity in right precuneus and IPS. So this area that was very close to the one observed during univariate analysis. For pseudo-word reading, we observe shifts in task-related activity in the right insula and central operculum. So it seems that RTMS of left PSDG modulates task-specific activity outside the specialized reading network in domain general areas associated with cognitive control and general processing demand. And such changes are highly task-specific, are different for words and pseudo-words. So overall, facilitatory RTMS effects in people with dyslexia are underpinned by task-specific changes, likely reflecting decreases in domain general areas, which points towards the role of such domain general areas under increased task demands in people with dyslexia. And maybe this also suggests that targeting such areas with facilitatory neurostimulation could be used to aid treatment or training protocols in such impairments. Now, in the second part, I want to move to targeting domain general brain areas to map plasticity across networks. So this is data from an fMRI study led by Ole Numsen from our lab, where we investigated domain general network interactions across different cognitive domains. So we focused on the inferior parietal lobe here, an area that is part of the default mode network, so this task negative network, but this area is also associated with different key domains of human cognition, language, attention, and social cognition. And based on tasks from these three domains, we were able to distinguish two regions in both hemispheres, an anterior one and a posterior one. And now we found that the functional connectivity profiles in these subregions differed and were highly domain specific. So we had differences in the connectivity profiles for attention, semantics, and social cognition with respect to its connectivity with the major brain networks in the rest of the whole brain, some of which are also described at rest. And this shows that even though we have a domain general area here, its connectivity profiles are modulated based on the specific task requirements. And now the next step is to target these areas with neurostimulation and see if the changes in the modulatory effects are also task-specific to better understand the task general versus specific contribution of these subregions. So this is work again led by Ole Numsen and Katie Williams from my lab, where they investigated task-specific effects of network stimulation in the default mode network. And this is work in progress, but some preliminary results suggest that the induced electrical field strength in the IPL is strongly correlated with the behavioral modulation induced by TMS in a highly task-specific manner. So here you can see the behavioral neurostimulation effect in the semantic task that we used, that it strongly depends on the induced electrical field strength in the left posterior IPL subregion. So there was a correlation between the induced electrical field strength. The stronger the induced electrical field, the faster the TMS-induced modulation of the semantic response speed. So the stronger the facilitatory TMS effect. These effects were highly task-specific, so selectively for word relative to pseudo-word processing, and they were strongly domain-specific, so only observed in the semantic task relative to attention and social cognition, and they were also specific for the subregion. And on the long run, this may have potential therapeutic relevance because we could optimize the neurostimulation protocols, so the targeting and the dosing of a specific protocol for the respective subregion and for the process of interest that may be impaired in a certain disease. So finally, I want to talk about domain general network interactions in the aging language network, because we know that many of our patients are of older age, and aging is associated with general cognitive decline and large-scale network reorganization, and it is debated whether such reorganization is compensatory or reflects dedifferentiation. So work led by Sandra Martin from my group shows that the increased interaction in domain general networks in the aging brain is associated with better behavior in older adults. So using a verbal fluency task during fMRI, Sandra showed that older adults show generally increased task-related activity in domain general areas, including midline structures. Older adults also show increased task-related interactions between domain general areas and networks. So here illustrated for the right insula of the multiple demand network and default mode regions. And older subjects show increased interactions within the domain general multiple demand network to be associated with better general behavior outside the scanner in executive tasks. So this shows that some of the observed reorganization patterns appear to be beneficial, appear to be compensatory in the aging brain. Using more advanced analysis, Sandra could further show that older adults show increased facilitatory network interactions, which are associated with better verbal fluency behavior, so also seem to be relevant for language processing. So this is data from an ICA and correlational PPI analysis to investigate changes or differences in network interactions really between old and young subjects. And these age-specific differences show an increased facilitatory network interactions between usually anti-correlated networks. So increased facilitatory network interactions between task-positive and task-negative networks. These facilitatory network interactions predict better verbal fluency selectively in the aging brain. So they seem to be of behavioral relevance, which again shows that some of the observed changes of the reorganization profiles are adaptive. And this suggests that we may target such domain general nodes with facilitatory neurostimulation to aid language recovery after brain lesions. So to summarize, what do we know about flexible redistribution across networks after neurostimulation? Perturbation of key language nodes results in strong remote effects, not necessarily at a stimulated area, and often associated with a shift towards domain general networks. Compensatory reorganization in domain general areas may help to maintain task processing at a high level. Facilitatory neurostimulation then may decrease domain general network contributions, likely reflecting less task effort. Domain general networks also show specialized contribution to different cognitive key domains as observed in the default mode network. And targeting domain general networks yields domain-specific modulation profiles as observed for the semantic task in our preliminary analysis with the E-field modulation. Domain general network reorganization also contributes to language performance in the aging brain, and some of these nodes may be good target candidates for facilitatory neurostimulation to aid recovery. With this, I'm at the end of my talk. Thanks for your attention. I wanted to have a job announcement here, so I'm will be looking for a postdoc soon in a project on network reorganization with concurrent TMS fMRI. If you're interested, please approach me. So that's it. Thanks for your attention.